Hey everybody, Mr. Grove here. Today we're going to talk a little bit about cell theory and the history of the discovery of cells. Cell theory as we know it today is all living things are made of one or more cells, and all cells come from existing cells, and cells are the basic units of life. But this was really kind of a compilation of many people's scientific work and discoveries. And in particular, these five guys that we'll talk about today had a large part in forming what we now refer to as cell theory. So the first was an English scientist, Robert Hooke was his name, and he was way back in the 1600s, so around 1665, and was the first to identify cells. He was looking at cork under a very primitive microscope, basically just some lenses, 3 lenses that were spaced apart. And he noticed that they looked like little kind of rooms, empty rooms. He described it as what monks would live in a monastery, kind of like a dorm room today. And so that's where the name cell came from. We would probably think of this more like the terms for a prison cell, but Hooke thought it was more like the monastery dorm rooms. So he was not looking at living things, and so really he just saw the empty space inside the cell wall. And that was Robert Hooke. If we fast forward about 10 years later, a Dutch scientist who actually was in the commercial world was Anton Levin. And he actually was using his primitive microscope that was a little bit better than Robert Hooke's. And he was a cloth trader, and so he was going to try and look at cloth under the microscope. And he noticed that there were animals in like a drop of water, and noticed cells in much more vividness and complexity because he was looking at living things. And so that was Leeuwenhoek's discovery. Later, a guy by the name of Matthias Schleiden, came, and he was studying plant cells. And so if you notice, about 150 years has gone by, and so it really took a long time to get traction to move forward this concept of the cell theory. And so Schleiden was studying the plant cells. He was a German scientist. And then at the same time, a year later, Theodor Schwann also concluded that not only animals but plants, so therefore everything must be made of living cells. And that kind of started our cell theory, as our first point says. But Schwann also was erroneous in saying he kind of thought that the cells could just spontaneously occur. And that seems a little silly for us now, but back then, people would see a piece of meat and then all of a sudden there'd be living things like maggots on it. And how did that happen? It must have been spontaneous. And so it wasn't until about 15 years after Schwann that another German scientist, Rudolf Verchow, proposed that cells have to come from other living cells. And so Virchow is a really interesting guy. He also did a lot of study on blood work, and he has some of our earliest scientific studies on cancer cells and leukemia. And so it was after he proposed that the cells have to come from living cells that helped kind of set the foundation of all of our cell theory. And so now we know today that all living things are made of one or more cells, that all cells come from existing cells, and that cells are the basic units of life. And that's our cell theory. Another aspect of cells is going to be their size. And so, how big does a cell have to be? Well, it's got to be big enough to hold all of the vital organelles and all of the vital components. So the DNA and the ribosomes and all the organelles of a eukaryotic cell. But yet, there's a size limit, because if we get too big, then what happens is we are not able to be as efficient. And so that's where some mathematical principles come into play: surface area and volume. So if we have 2 cubes, so like this cube here, if it was 30 by 30 by 30 on one side, would have the same volume as 27 of these cubes that were in kind of like a little Rubik's cube here if they were 10 by 10 by 10 on each side, and we had 27 of them. So the volumes would be exactly identical. But the surface area, so we get surface area by taking the area of one side and then adding all those sides together for the surface. So in this case, we'd take 30 times 30 and then take that times 6 because we have 6 sides. And that would give us a surface area of 5,400. On this side, our surface area is going to be 10 by 10 times 6 for one little cube, and then times 27 for all 27 cubes. And so the surface area is going to be 16,200, basically 3 times as big if we just had a large giant cube. So the surface area to volume ratio would be significantly bigger for our smaller cells. And so that kind of limits us on how big a cell can be and still be efficient. So we kind of need to hit that sweet spot where we're big enough that it can hold all the necessities, but yet we don't want to get too big because that efficiency of taking nutrients into the cell and expelling waste out of the cell, if that's not as efficient, that cell is going to be less likely to thrive. So some of the examples in our body of some big and small and long cells. So some of the smallest cells you would have would be like your red blood cells. They have to be really tiny to get through some of those tiny capillaries in your body. Some of our bigger cells, eggs, like bird eggs, are oftentimes considered to be one cell. And so they have to be big and packed full of nutrients for the young embryo to survive. Some of our longer cells would be like our muscle and nerve cells, so these long fibrous types of cells. And so hopefully that helps you understand how big a cell has to be, but yet why it can't be too big in order to be effective at doing what it does. So hope that was helpful, and we'll see you next time.